Oh, okay, sir. Good afternoon, my dear students. So I am Dr. Kirani from Government Degree College, Siddipeta, Autonomous. Today we are going to discuss about the embryology topic. So up to yesterday, we'll discuss the nitrogen metabolism, and today we start the embryology. You know that the embryology means the study of development of embryo is known as embryology. Simply, what is embryology? The study of development of embryo is called as embryology. So in the embryology, the father of embryology is Acharya Panchanan Maheshwari. So it is very important in entrance point of view. Who is the father of embryology? Acharya Panchanan Maheshwari. So in that, generally in angiosperms, the male reproductive party are the stamens. We know that in the flowering parts, almost so the stigma, style, stamen, ovary, corolla, calyx, thalamus, pedicel, these all are the some parts of the flower. So in that, today we are going to discuss about that the anther TS or structure of the microsporangium. In simple way, the structure of anther or microsporangium. So it is also known as stamen or microsporogenesis. So in that, this is the complete the structure of the anther parts, Nana. So almost in angiosperms, the male reproductive part are the stamens. So the stamens have filament and anther. Completely, the stamens have the filament and anther. So in that, the anther is a present at the end of the filament. So when you observe the complete imagine the flower parts, so the anther is the present at the end of the filament. So in that, the antheres are of two types. One is the dithecous anther and second one is the monothecous anther. So what is the dithecous? Dithecous means anther having two lobes, that is the dithecous anther. So majorly it is found in many plants. So the anther, that means the sepal wall divides the anther into four lobes. Just to we'll seeing at that time. So yeah, that is the dithecous. It having the two lobes only. So in dithecous anther, the sepal wall divides. So it will show anther into the four lobes. It's clear, Nana? So next, the anther having the only one lobe. So that is called monothecous. So this is found in family Malvaceae. It is the primitive families almost. So only one sepal wall is found. So it divides the anther into two lobes. Almost two lobes. The anther, it is completely, the septal wall divides the anther four lobes. That is the dithecous. In monothecous, so only one septal wall and it divides into the anther into two lobes. So the anther is known as a microsporangium and the ovule is known as a megasporangium. So with that, this is simple, just one or two introduction point of view. That is the anther or stamen microsporangium. So when we entering into the structure of anther or microsporangium, almost so the stamens are microsporophyll are the regarded as the male organs of the flower, Nana. So each stamen, it consists of the three parts. One is the filament, anther and connective part. So filament part and anther and connective. So in that the filament is the slender stalk of the stamen. So and the anther it is the expanded head. So it is borne by the filament at its tip part. Each anther consists of usually of two lobes, as I told you that. So it is connected together by a sort of the midrib. So that is the connective part. Each lobe of the anther, it contains the two pollen sacs or two microsporangias. So thus there are the four chambers in each anther. But in many cases, there are only two and sometimes even only one also. So in China rose or lady's finger, we will observe only one chamber. So within each pollen sac, there is a fine powdery or granular mass of the cells we observe. So that is called pollen grains, or it is also known as a microspores. Microspores are pollen grains, both are equal. So in that, pollen grains are produced in large quantities in the pollen sacs. Okay. So when we entering into the microsporangium or pollen sac, the anther consists of the 4 parts, Nana. So in that, the microsporangium or anther consists of the 4 layers. One is the epidermis, and second one is the archisporium. So in simple way, broad thing, one epidermis, and second part, completely inner part, is the archisporium. And archisporium also divided into 2 layers. One is the parietal tissue, and second one is the sporogenous tissue. So in the parietal tissue, it is divided into 3 sublayers: endothecium, middle layer, tapetum. Don't be confused. So completely, we discussed today only the anther structure. Anther parts, one is the epidermis, and the inner part is the archisporium. So archisporium consists of the 2 layers. The outer part is the parietal tissue, and completely inner part is the sporogenous tissue. So in that outer part, parietal tissue consists of the 3 sublayers: endothecium, middle layer, and tapetum. So as per same, and the internal part, the sporogenous tissue consists of the pollen mother cells, pollen tetrads, and pollen grains. So this is completely the flow chart of the anther structure. Now we'll discuss one by one in the all the 4 layers. So in a flower, the stamen is considered as the male reproductive organ. So as I told you, each stamen, it consists of the filament, connective part, and anther. Okay, filament part, connective part, and anther part. So the anther may be, it is either monothecous or dithecous. So a monothecous anther, it consists of the 2 lacules or 2 sporangia. Okay, the monothecous anther, it consists of the 2 lacules. That means one is septa is divided into 2 parts and jeppunam. So in the 2 lacules or 2 sporangias, so it is said to be a bilacular or bisporangiate. Okay, and as per same, a dithecous anther, so it consists of the 4 lacules or 4 sporangias. So it is said to be a tetralacular or tetrasporangiate. You can observe here in the diagram. This is the complete the anther structure. So the structure of the anther, in that the monothecous and dithecous. So it is completely the dithecous anther, Nana, the transfer section of the tetrasporangiate anther. So it's showing the various tissues. That is the anther structure. So in this, almost the each stamen, so it arises a small papillate outgrowth of the meristematic tissue from the growing tip of the floral primordium. So we know that it grows actively and soon gets it to differentiated into an apical broader portion. In that, the anther and the lower slender part, so that is the filament, the completely broader portion in that anther and slender part and the filament part. So just to, you can imagine the complete the flower part. So in that flower, the stamen, filament, connective, and slender parts all are, we'll observe in the flower anatomy. So the cross section of a very young anther, it consists of a homogeneous mass of the meristematic cells. So they are surrounded by an epidermal layer. Further, it growth of the anther makes it 4-lobed. So this is the 4-lobed part. You can observe microsporangia and all the microsporocytes, that is the connective part, and the epidermis, the outer part is the epidermis. So in that, a cross section of a young anther, it consists of a epidermis. So in simple way, epidermis, endothecium, middle layers and tapetum parts. Okay. So in that epidermis, almost the epidermis is the outermost protective layer, Nana. Okay. So completely it look like a unicellular cells. They are arranged in the compactly arrangement of the cells. So outermost protective layer of the microsporangium. It consists of a single layer of cells. So it does not have intercellular spaces. So see here, almost the outer part just to look like thick cells, you will observe single layer of the cells. Okay. So the epidermal cells in a mature anther, it become a stretched. So after maturity of the anther, so the cells become a stretched and some much flattened also. Okay. So in that some of the epidermal cells are larger in size and it is called that stomium. Just to in the diagram, the two this parts, Nana, the in between the two lobes. So this way and this part, these two parts, the cells just look like a large cells will observe in the two curves plot. So that is called that stomium cells. So why the stomium is presenting in the anther? Almost it helps in the dehiscence of the anther. Completely at the maturity condition of the anther. So the stomium cells, they can absorb the moisture or water. So it completely helps in the dehiscence of the anther. That means when releasing of the pollen grains. So at the during the maturity condition, it will helps in the dehiscence purpose. Okay, that is the epidermis. Epidermis is the outermost protective layer. So it does not have any intercellular spaces. So completely the cells in the mature, it becomes the stretched condition. The cells completely the epidermal cells only two places. Whenever the four lobes, we observe that the completely in between the two lobes, the larger in size cells presenting in that, the cells are called stomium cells. So they are different from other epidermal cells. Okay. So the stomium cells, they can help in the dehiscence of the anther. So in that, completely the second part is the endothecium. So this is formed by a single layer of cells. So this is the epidermis and second layer. This one or two layers are the endothecium. The endothecium means, so beneath the epidermis, it is a single or two layer cells below the epidermis. These cells are large in size. So when compared to the epidermal cells, the endothecium cells are large in size and do not have any intercellular spaces also. So these cells made of a callose or alpha cellulose chemical substances. So this is 2 years back, this is the entrance point question. The endothecium cells made up of a callose or alpha cellulose substances are presenting in the endothecium. Okay. And the third part, that is the middle layers, Nana. So it is found below the endothecium. This is completely in between, beneath the endothecium. 1 or 2 layers is the middle layers. So this layer is made up of 1 to 3 layers. So 1 or 2 or 3 layers of the cells, and it stores the starch completely. It will storing purpose cells. It will be stored the starch substances. So that is the middle layers. And at the same time, these middle layers, they can degenerate by that time. So that means meiosis takes place in the microspore mother cells. Whenever the microspore mother cells undergo the, it will take the meiosis divisions, the middle layers, middle cell layers, they will be degenerating at that at the situation. Okay. And the fourth one is the tapetum. So in the tapetum, almost the tapetum is the innermost layer of the anther wall. So beneath the complete, these layers are the tapetum cells. The tapetum, it is the innermost layer of the anther wall. It surrounds the microsporogenous tissue. So these tapetal cells are useful in the nutrition of the microspores. Okay, so completely, what is the main function of the tapetum? Tapetum cells is for nutritive purpose. Okay, so the tapetal cells are useful in the nutrition of the microspores. This is also entrance point question. Okay, Nana, the tapetal cells are useful in the nutrition of the microspores. The tapetal cells have the dense cytoplasm and large nucleus. So completely based on the development to tapetum, the tapetum is of two types. Just to we'll discuss that. It is the innermost layer of the anther walls, and it is completely surrounds the sporogenous tissue. So the cells containing the dense cytoplasm with prominent nuclei. Usually tapetum consists of single layer of cells. So as the tapetum completely surrounds the sporogenous tissue, so major part of it is derived from the parietal cells and a small part developed from the sporogenous tissue. Just to, in the flow chart, this is the sporogenous tissue. Almost major part is the parietal tissue and little part of the sporogenous tissue. So that is completely in the tapetum part. Tapetum transport the nutrients to the developing sporocytes. So the tapetal cells are pigmented and it is red-brown in apple or violet in the anemone. Okay, so completely it is different from plant to plant. So the tapetal cells are either just to, it is pigmented enough for that. Completely the main function of the tapetum is it is nutrient to the developing sporocytes. So on the basis of behavior, there are two types of tapetum we observe. So in that first is the amoeboid tapetum and second one is the glandular tapetum or secretory tapetum. First, amoeboid tapetum. So in the amoeboid tapetum, the inner and radial walls of the tapetum and the breakdown due to the action of hydrolytic enzymes and their protoplast penetrates between the pollen mother cells and developing the pollen grains. So in simple way, amoeboid tapetum means the amoeboid cells are formed from the immature tapetal cells. In the simple way, so what is the difference between amoeboid and secretory? Amoeboid tapetal cells are formed from the immature tapetal cells. For example, some lower dicotyledons and some lower monocotyledonous. Immature dicotyledonous and immature monocotyledonous. And at the same, the secretory or glandular tapetal cells, they are developing from mature cells. Completely mature cells of the tapetum is known as secretory or glandular tapetum. So, for example, all dicotyledonous, that means mature dicotyledonous or many dicotyledonous and higher monocotyledonous. Developed monocotyledonous, all are belongs to the glandular tapetum in simple way. But what is the development of the amoeboid tapetum? So completely the inner and radial walls of the tapetum, it break down due to the action of the hydrolytic enzymes and their protoplast, it will penetrate between the pollen mother cells and developing pollen grains. So after interaction, they fuse with each other and it will form a mass of tapetal periplasmodium. Okay, so this periplasmodium or this tapetal plasmodium remains associated with the pollen grains till their maturity. So some examples like amoeboid tapetum, that is considered as the primitive type, as I told you. So it is also called periplasmodial tapetum, the alisma, tridenscantia, typha, sagittaria, potamogeton. So these all are the best examples of the amoeboid tapetal plants. Alismaceae, tridenscantia, typha, sagittaria, potamogeton, almost all the hydrophytic plants. So immature or primitive plants. So they all are belongs to the example of the amoeboid tapetum. And second one is the glandular tapetum or secretory tapetum. So as I told you that completely they are the higher monocotyledonous or developed plants. Many of the dicotyledonous and higher monocotyledonous also they are the best example of the secretory or glandular tapetum. So in this, the development of the glandular tapetum is the cells of the glandular tapetum, it remains intact throughout the microspore development. So just two other diagrams. Oh, not in that. So they secrete their substances from their inner faces. The secretory tapetal cells are thin and they will possess almost all cell organelles like mitochondria, plastids, and dictyosomes. All are presenting in this tapetal cells. So that's why just before the pollen mother cells undergo the meiotic divisions, so the walls of the tapetal cells become thick and there is considerable increasing in the number of ribosomes, and the pro-ubis bodies also presenting with the completion of the pollen development. Pro-ubis bodies, they are passed into the antherlacule from the tapetal cells and they are now called the ubis bodies. So ubis bodies are presenting in the glandular tapetum. This is entrance point question. Ubis bodies, so they are presenting in amoeboid or secretory or glandular. So both ubis bodies are presenting in the matured cells. So they developing from mature cells of the tapetum is the glandular tapetum. So they have consist of the some ubis bodies and they coated over the pollen grains. So almost, as I told you, the examples higher monocotyledonous and many dicotyledonous. So almost what is the functions of the tapetum? The nutrients are transported through tapetum to the sporogenous tissue, and the tapetum is involved in the synthesis of the callus. It will release microspores in a tetrad by degrading the callus wall. So the tapetum plays an important role in the formation of the exine. So almost the pollen grain, it will covering with complete sporopollenin. So in that, the tapetum plays an important role in the formation of the outer layer of the pollen grain cell wall, that is the exine. And in the pollen kit, so lipids and carotenoids, pollen kit is formed by the tapetal layer. It is a insect attractant and it will protect pollen from the ultraviolet cells also. Ultraviolet rays. Okay, not only the nutritive purpose, it is involved in the synthesis of the callus. So it releases the microspores in the tetrad by the degrading of the callus wall, and it is also help in the formation of the exine layer, and it is also form insect attractant, or it will protect the pollen from the ultraviolet rays. Okay, so this is completely the anther structure, Nana. So completely the development of microsporangium or pollen sac. It will containing the epidermis and endothecium, middle layers, and tapetum. So tapetum is amoeboid tapetum and secretory tapetum. And what is the in the development of the pollen grain or microsporogenesis, the microspore mother cells. Sir, it will not move. Okay. Okay. So the microspore mother cells, they will undergo the meiotic divisions and it gives rise to the microspore or pollen tetrads. So this is the first slide. I'll show once again. In the sporogenous tissue, completely it will form the pollen mother cells. So after that, the pollen mother cells undergo the meiotic divisions and it gives rise to a microspores or pollen tetrads. So almost 4 cells are formed by the pollen tetrad. In that, the microspore or pollen, that is the uninucleate, Nana. Almost, So even in the tetrad condition, the microspore or pollen grain is the uninucleate, single celled and globular structure. So this is almost what is the main structure of the microspore. Microspore is or pollen grain is the uninucleate, single cell and globular structure. So in that, the outer layer of the microspore is made up of a pectin and inner layer is made up of a cellulose. So next we will discuss that what is the main composition of the pollen grain. So in that exine, all are coming in that. But in the broad way, the pollen or the microspore, the outer layer consists of the completely sporopollenin. So it checks the natural decay of the pollen grains. So completely thousands and 100 years ago, we will observe in the fossil formation, we will observe the pollen grains because of it will consist the sporopollenin. So it's very hardening outer layer. So the microspore outer layer consists of the sporopollenin. So it checks the natural decay of the pollen grains. So it is due to the sporopollenin that the pollen grains get preserved during the fossilization process. Okay, so for that purpose, almost the outer layer has pollenicate that helps in the attraction of the insects also. In the pollination purpose, it have some designs or nourishment of the outer layer will observe in different plant pollen grains. So that for that purpose, the outer layer has the pollen kit and this helps in the attraction of the insects. So in the flower, the stamen, almost we will discuss that, considered as the male reproductive organ. It consists of filament, connective, anther, so dithecous anther and monothecous anther. So in the archisporial cell divided periclinally, the epidermis, endothecium, middle layer and tapetum. This is the epidermis, outermost single layer, it compactly arranged and usually protective in function. So epidermal stomata was reported in Alangium. And endothecium, so for example purpose, I'll repeat again. The cells of the endothecium are radially elongated and it will show the fibrous band. The fibrous bands are made up of a callus and arise from the inner tangential walls. So usually some fibrous bands are U-shaped. In other way, some of C are U-shaped also will observe the fibrous bands. So the fibrous bands are hygroscopic in nature and in the endothelial cells help in the dehiscence of the anther at maturity level. So because of the presence of fibrous bands, this layer is otherwise called the fibrous layer also. So the endothecium is also known as the fibrous layer. Okay, so it is usually single layer. So only in exception of coccinia. Coccinia lo double layer untundi. Almost majority of the plants, the fibrous layer is usually single layer. Okay, so below the endothecium, 2 to 3 layers of the cells, middle layers. So they are ephemeral and they become crushed by the early meiosis in pollen mother cells. So this is act as a storage centers for the starch. They are food preserving part in the anther is middle layers. So almost all the starch products are preserved or stored in the middle layers of the cells of the anther. So this is the tapetum. Almost time completed, I think. Remaining 3 minutes more. If you have any doubts, please ask me. And tomorrow we'll discuss that I'll show some other diagrams like just to flower pots, other flower pots. Just to wait. Hello, dear students. Any doubts regarding the introduction of the embryology or anther structure. So in that almost in the embryology, just some history of embryology, I will show some slides.